Hello and welcome to today's webinar. We will be reviewing a scalable and cost-efficient method for targeted enrichment of long fragment in NGS. Today, we have two presenters that I want to introduce. We will have Professor Andre Franker, Director of the Institute of Clinical Molecular Biology at Kiel University. Dr. Franker has a strong theoretical and methodological background in human genetics and bioinformatics. The main focus of his research is on complex chronic inflammatory disease, such as inflammatory bowel disease, psoriasis, and sclerosis cholangitis. On the other hand, our second speaker is Tim Steyert, who is currently a PhD candidate at the Institute of Clinical Molecular Biology in Kiel. His research focuses on optimization and the development of techniques in the field of second and third generation sequencing. Thank you both for this wonderful presentation.
Yeah, thank you for the introduction and thanks for joining our webinar today. So as announced, we want to present to you today a method for targeted enrichment of log fragments for long-read sequencing. But before Tim will walk you through the method and the details of the protocol, I wanted to give you some background on the rationale of long sequencing. I also wanted to outline some challenges that you may have with long-read sequencing, and I want to give you two examples. Long-read sequencing has been titled Method of the Year by Nature Methods in this year's January issue. Long reads have several advantages, and most prominently they can span regions that are very complex. And this may help you to better detect, for example, structural variation, such as inversions or insertions, for example, complex chromatopsis, or complex rearrangements, repeat expansion length variations, highly polymorphic regions. But long reads may also be quite useful, for example, if you want to analyze full-length 16S ribosomal RNA. Additional clinical applications are sequencing of genes with a pseudogene or detection of modified DNA bases, including methylation. They can also be very helpful if you want to analyze complex regions like the HLA or the killer immunoglobulin receptor. Here, long reads may help to infer haplotype phase. Also, full-length RNA transcript analysis to detect alternative splicing events is of interest. Or applications, of course, where a portable device is of an advantage if you work with the Oxford Nanopore system, for example. So let me just give you two examples from our own research where long-read sequencing is beneficial. Firstly, we are interested in human blood groups. And you may know that there are many more blood groups than just ABO. And it just happens that the genomic analysis of two clinically very important blood groups, rhesus and MNS, is not that straightforward with short reads. And here, for example, you get an impression of the complex architecture of the MNS blood group logos. It is about 280 kb long. There are, of course, many SNPs. There are genetic hybrids. We have gene duplication events, excessive homology. So here we really need long reads to decipher the exact genomic sequence here for an individual. And it's similarly complex for the rhesus locus. Secondly, we are very much interested in mucins, and particularly the MUC2 gene locus. As you see in this illustration, the central exon of MUC2 is very long and repetitive, and therefore it cannot be easily resolved with short reads. In fact, mucin genes are often excluded during or after the variant calling step in human whole exome and genome analysis. Nevertheless, mucins are biologically very interesting, as the mucus formation on our gut epithelial cells, for example, protects us from the environment by keeping, for example, bacteria away from the cells. An intact mucous barrier is important for health, and a thinner layer has been described, especially in patients with inflammatory bowel diseases. And that's what we're interested in, and that's what we're doing research about. So if there would be a technology that could facilitate an accurate and high throughput analysis of the MUC2 gene variation in a larger set of cases and controls, we could definitely better test for an implication of this gene in disease. So I mentioned the benefits of long-read sequencing, followed by two examples. Still, challenges remain. For example, the accuracy per third generation sequencing read is lower still than in short read sequencing. Next, the cost per base is also still higher than in short read sequencing. And also the sample throughput is lower. It is not that well established in clinical labs, meaning that training and new workflows are often required. Bioinformatic approaches are less mature often than for short read sequencing. And input DNA should ideally be in long fragments, so high molecular weight DNA, which can be a challenge for standard automated lab DNA isolation methods. So, targeted enrichment approaches are scarce. So new approaches for targeted enrichment here for particularly long-read sequencing would be very useful. And with this, I would like to hand over to Tim, who will now introduce you to a really good approach that we have established here in-house.
Thank you, Andre. Also, thank you, Clotilde, for your nice introduction. After Andre explained the background of everything, I would like to switch gears a bit and give you an overview about the protocol itself, point out the time savings, also state the reagents and equipment required for the preparation, as well as show you some peer-reviewed outcome data of the protocol. Afterwards, I would like to also talk about the challenges, which is mainly sample quality as well as sample quantity, the applications of the whole protocol. I would like to show you how we can achieve haplotype phasing with our protocol, how we can bridge the gaps, elucidate structural variation, and resolve difficult genomic regions, and how we are going to apply this for our blood group genotyping research that Andre already mentioned. In the very end, I would like to also talk about the interfaces, which are at the moment Pacific Bioscience or PacBio, as well as Oxford Nanopore Technologies. This is the technical situation I was in before I started my PhD thesis. For next generation sequencing, both amplicon-based and hybridization-based techniques were readily available in a high throughput fashion. However, for third generation sequencing, only amplicon-based protocols were really high throughput capable and hybridization-based protocols were not. The reason why hybridization-based protocols were not can easily be seen on the complexity of the whole workflow. First, one needed to fragment high molecular weight DNA, perform a cleanup, an end repair, an A-tailing, and adapter ligation reaction, then another cleanup, then amplify all the fragments in the first PCR reaction, perform a cleanup, then perform a very labor-intensive gel electrophoretic size selection, another cleanup, then enrich the fragments with your custom bait panel, perform the second round of PCR amplification, another cleanup before one finally could enter the long-read sequencing workflow. As you may see, we really had to come up with a solution in order to increase the high throughput capability of that protocol to prepare more samples as it was required by our research question. We identified the electrophoretic size selection as being the most cost and labor intensive step of that whole protocol. constituting the throughput bottleneck, also preventing the automatization of the whole protocol, and because of its low yield, being the requirement for a second PCR amplification, and therefore also reducing the sequencing quality in terms of duplicates and read length. Let me give you the bigger picture, as we are doing something differently as compared to classic next generation sequencing enrichment techniques. So as you may know from next generation sequencing enrichment, your fragmentation is usually done via ultrasonication or enzymatic fragmentation. Then from these fragments, you prepare your short read sequencing library. You perform then the hybridization with a readily prepared library using your custom panels before everything is going to be sequenced via next generation sequencing. In our case, we perform the fragmentation with the Covarius G tubes, and then there's an inverse order because we perform the hybridization with the barcoded fragments before preparing the actual library. The advantage of that approach is actually that you, with your enriched fragments, can flexibly decide if you're going to sequence everything via PacBio or Oxford Nanopore. And then with your enriched fragments, you can decide which workflow to enter. These are the modifications that we've done to the standard enrichment protocol in order to improve the per sample economy and also increase the sample throughput of the whole protocol. We could get rid of the before mentioned gel electrophoretic size selection by replacing an already existing cleanup through a bead-based size selection that we got from literature. By doing so, and because of the higher recovery rate of that cleanup, we could get rid of the first PCR amplification that was done sample individually. And also, of course, we could get rid of the associated cleanup steps. In terms of quality, we also ended up better off in the very end because of the higher yield, the higher recovery rate of that bead-based size selection. We had to perform less PCR cycles in total, ending up with a longer fragment length and a lower duplicate ratio. This is the streamlined version of the protocol that we published in 2022 that only consisted of a fragmentation, the size selection that was done via beads, so really scalable, the end repair, the A-tailing, the adapter ligation, a single cleanup in the whole protocol, then the actual enrichment with the custom panel, then one single PCR reaction that was done in a pooled manner, then a last and second size selection before the desired long-read sequencing workflow is entered. When compared to different protocols from literature, the efficiency gain that we could achieve with our protocols really become obvious. So we don't need 16, 14 or 51 days to prepare an exemplary set of 96 samples. In our protocol, these samples are just prepared in a little bit more than 2 working days. On the 3rd working day, the desired 3rd generation sequencing library preparation of your choice can be started, as well as the sequencing run itself. In terms of multiplexing, with our protocol, we can achieve 16x of multiplexing, while compared to just 8x multiplexing in another protocol. Moreover, also the requirement for capital investment is reduced because we don't need any device for electrophoretic size selection to prepare samples according to our protocol. It's simply a centrifuge, SPRI beads, as well as a thermocycler. This is an overview on the reagents and equipment required for performing the capture enrichment. For the fragmentation, you would require the Covarius G tubes in combination with a standard mini centrifuge. The size selection is done with Beckman, Coulter and Pure XP beads. You would need to exchange the buffer of these beads with the below mentioned chemicals. So it's lithium chloride, PEG 8000 and tris with a pH of 8.5. And with that exchange buffer, these beads are then capable of performing the size selection in the required range of fragment sizes. The entry-pair A-tailing and adapter ligation is performed using the IDT-XGen DNA library prep MC kit, as well as IDT custom DNA oligos. You would need these oligos for barcoding your fragments, and you would need a respective amount of these barcodes depending on the size of your target loci and depending on how many fragments you want to pull together. The hybridization enrichment itself is performed with the IDT-XGen hybridization and washgate version 2, as well as your custom hybridization panel, and you would require one IDT custom primer for the final amplification. The long-range PCR is performed using the Takara LA-TAC DNA polymerase in its hot start version. Also, you require a thermocycler for all the steps onwards from the entry-pair A-tailing and adapter ligation. This is some peer-reviewed data from our publication last year. We hybridization enriched 2.2 megabases of blood group loci in a 16x multiplexing level from an initial input of 1.5 microgram per sample, achieving greater than 7 kilobases in fragment size and greater 6 kilobases in hi-fi reads. And due to the repetitive sequencing of those fragments, we were able to achieve a mean accuracy of 99.98%. Also, what you can see here is the distribution of reads between samples, and maybe with the exception of one sample, the samples achieved relatively even in terms of read number. And the good thing to say is also that the mean target coverage was almost 143x. Also, we were able to achieve a very good output for our sequel2 sequencing system, a low duplicate ratio, and a on-target rate of 2/3. We also included some very challenging to sequence paralogous loci within our panel design. And what you can see here is the comparison between some normal loci of equal sizes and these paralogous loci. You can see here that the normal loci are of course sequenced better, so the distribution of coverage is more even as in the paralogous loci. However, the paralogous loci are also covered quite well, and with that data you can do whatever analysis you want to perform. This now leads me to the challenges of our protocol, and the biggest challenge is sample quality. Unfortunately, there's no way of ligating fragments back together, so if there are no long fragments in your sample, you cannot sequence long fragments from your sample. But what we can achieve with that bead-based size selection is that we can get the most out of your sample. What you can see here is a DNA ladder that has been treated with our size selection beads, and you can see that after the size selection, the high molecular weight fraction is significantly enriched, and all the smaller fragments of that ladder are gone. The second challenge is sample quantity. Our protocol works in a range of 1.5 to 5 microgram of high molecular weight DNA input. However, in order to account for sample differences, as well as applying a safety margin, we would recommend you to use 3 micrograms of input DNA. 1.5 micrograms of input DNA may sound much to you in the beginning. However, if you would correct for the fragment size and just use the same amount of molecules for an exemplary 500 base pair fragment size as used in short-read Illumina sequencing, you would end up with just 75 nanograms, which is actually quite comparable to what's out there on the market. Also, as previously stated, we have successfully multiplexed up to 16 samples with 200 nanogram each for hybridization. So why would you do long read sequencing at all? This slide is meant to show you the difference of some genotyping technologies such as array chip genotyping, next generation sequencing, as well as long read sequencing for a sample with a diploid genome, which is indicated by the presence of 2 reference alleles here. The array genotyping SNP would only resolve you some predefined positions, that are here indicated by the allele defining SNPs that you all considered in the design of your respective array chip. However, with these positions, it's very difficult to really resemble back your reference alleles, as you only get the information whether your SNPs are present in a heterozygous or in a homozygous configuration. Next generation sequencing does also resolve you some genomic context of your respective allele defining SNPs. However, also these SNPs can very rarely be put back together into one phase resembling your reference alleles. This is because the classic short reads does only span a few hundred base pairs in length, and your heterozygous SNPs that are required for phasing back your reference alleles are very rarely located in such a close proximity to each other. This is resolved by the long reads of third generation sequencing applications, since these reads have many thousand base pairs in length, more reliably spanning the distance between these allele defining SNPs. Moreover, the advantages of phasing back the reference alleles are also comprising the differentiation between in cis or in trans compound heterozygous mutations, as well as full-length RNA sequencing and the isoform identification, the detection of structural variation that is done more easily with such long reads, as well as haplotype reference allele assembly. This is an example of how well long reads can be used to phase the haplotype in case this is required for your application. On the right, you can see a screenshot of IGV, where 3 reference samples, the Ashkenazium trio, the mother, the father, and the son, were hybridization capture enriched and sequenced with a PacBio Sequel II machine. The haplotypes could be resolved over the whole ACKR1 gene, which totals in 4 kilobases in length. You can nicely see that the son inherited haplotype 2 of his mother, as well as one of the identical alleles of his father. Another advantage of capture enrichment with long fragments is what we call bridging the gaps. Sometimes, through repetitive sequences or sequence paralogies, with capture enrichment for next generation sequencing, you will have genomic areas that are not unique enough to design capture probes against them. Such an area can be seen below here, with the blue bars indicating the individual capture probes that we use to enrich our targets. For a specific area of the GIP-E locus, for this 5.6 kilobases gap that you can see, we could not design any capture probes against the target sequence, since paralogies to other areas of the genome existed. The advantage of long fragment capture is now that just a few baits at the very flanks of the targeted fragments are enough to pull them down in the enrichment procedure. So you can see that despite no probes are targeting the whole area here, the coverage is decreasing, however it never gets zero. This is depicted here for the PacBio hybridization on the top and the Oxford Nanopore hybridization on the bottom track respectively. This is how you can unravel what's in these difficult to sequence areas with a high sample throughput. This slide illustrates why capture enrichment of long fragments is the optimal way to go when the detection of structural variation is of interest in your application. On the right-hand side, you see IGV tracks representing different approaches in sequencing of the RHCE locus. In the top track, you see the hybridization enrichment and subsequent PacBio sequencing. The middle track represents a PacBio whole genome sequencing approach, while the bottom track depicts Illumina short-read next-generation sequencing data. In the marked area, the short-read sequencing dataset misses some important information: 122 SNVs and 3 smaller deletions, as well as one very important 109 base pair insertion that is actually deterministic for a serological phenotype. All variations are observed and confirmed in the independent PacBio datasets with very long fragments, high mapping quality, and hence high certainty. However, due to output restriction of the Sequel II machine that we use for sequencing, only one read covers this specific area, which makes profound reasoning quite challenging. In contrast to this, PacBio hybridization enrichment data is highly covered and rock solid in terms of reasoning, and in addition to this, both haplotypes can clearly be separated and be resolved. This is another example for why long-read capture enrichment could potentially be the key for resolving difficult-to-sequence areas in your application. Here you see a screenshot from IGV with short-read whole genome sequencing as well as PacBio and Oxford Nanopore hybridization enrichment experiments. The sample sequence is the human genome 001, the reference sample of the Utah Mormon mother. The short-read track comes from the Illumina marketing dataset, the so-called Platinum genome, while both long-read datasets were sequenced at our institute. The light color of some reads implies that the mapping quality is 0, indicating that these reads are not uniquely mapped to a particular region, but they may also be mapped to another genomic locus. Only very few of the reads can be mapped uniquely, since in their alignment area there might be some unambiguous sequences present, allowing them to be anchored at these coordinates. Variant detection in areas with low mapping quality is particularly difficult and may be significantly biased by mismapped fragments. On the other hand, long and accurate PacBio reads have no problems anchoring to these unambiguous areas and thereby spanning and resolving difficult-to-sequence regions. Some Oxford Nanopore reads also not map uniquely to their genomic location. However, there are always enough uniquely mapping reads left so that the variation in this area can unambiguously be elucidated. To come back to Andre's introduction into the application to our specific blood group research question, here's a slide on the outcome of our previously mentioned method publication. For benchmarking the accuracy of our method, we perform testing according to the FDA Truth Challenge in a set of 7 correlated reference samples and achieved high accuracies with the benchmarking data over the whole enrichment area of 2.2 megabases. As 6 of the reference samples constituted trios, we also validated the Mendelian errors within these trios, yielding an even higher accuracy. Moreover, we performed blood group genotyping in a 9-patient cohort. 1 patient from this cohort was genotyped via NGS as well as via targeted PacBio sequencing. In 3 cases, we could refine or even correct the respective blood group allele reference calls determined by next-generation sequencing through the PacBio long-read data. The blood group phenotypes determined by area genotyping for the other 8 patients were perfectly concordant with the phenotypes determined by long fragment target enrichment. This leads me to the first of our 2 interfaces, which is Oxford Nanopore technology sequencing. On the left, you see a very simplified sketch on how this technology works. A mosoprotein pulls the fragment through a nanopore located in a biomembrane while the ion flow through the nanopore is being observed. Depending on the steric blockage of a nucleotide multimer in the pore, the ion flow changes and consequently the strand's sequence can be computationally reconstructed. As it has low entry barriers, a quick turnaround time, and good usability, this technology is especially relevant for smaller laboratories. This technology is a fast and easy way to enter long-read sequencing. The single sequencing reads are less accurate as compared to our second interface, but using the consensus accuracy of all reads, most research questions can be addressed. Pacific Bioscience sequencing, our second interface, shares the principle of sequencing by synthesis with NGS approaches. However, in PacBio's case, this is done in real time and for single molecules as compared to sequencing cycles and cluster sequencing in NGS. PacBio collapses the redundant reads of the same molecules to what they call circular consensus reads. In our case, a repetitive sequencing of our enriched fragments yielded greater than 99.98% accuracy with a target coverage of around 500x. This interface is the right to choose when high sequencing quality is required. With the launch of a new sequencing machine, also the previous throughput restrictions have been addressed. That's it from my side. Thank you very much for listening. Also, thank you, Clotilde, for the opportunity to present this webinar. I would be happy to answer your questions.
Thank you, Tim. Thank you, Andre, for this wonderful presentation. I have a few questions, and I think we're going to start with you, Tim. So, did you require any additional capture probe optimization for your target loci, or did you just use a standard panel?
Thank you very much for your question. Yes, indeed, we did require some optimization. The first level of optimization was done by IDT, as we specifically asked for a panel that is meant to do long fragment capture. Second, we needed to optimize the tiling of the probes and ended up with a 0.5x tiling. This means that 50% of our target bases are actually covered by the probes. And as these probes are 120 base pairs in length, that means that the first 120 base pairs are covered with a probe, the next 120 base pairs are not covered, and the next 120 base pairs are covered again with probes. And lastly, also, we needed some optimization with regards to the flanks. Long fragment capture works in a way that the coverage slowly builds up for the target area. So what we needed to do is we actually had to extend our flanks upstream as well as downstream of our target areas so that the coverage could slowly build up over these flanks, and then that we had our whole target covered, even the first and the last exons of our target genes.
Thank you for this answer. I have another question for you. Is there anything specific to consider when it comes to choosing the DNA extraction kit? We have many valuable old samples that we would like to sequence. What is the quality exclusion criteria?
Thank you very much for this really important question. So actually quite recently, we had to make the experience that we needed to exclude many samples that were sent to us by a partnering facility due to too poor quality. And this was really sad since these samples were very rare and precious. The exclusion criteria on our side is generally if a sample has more than 30% of its fragments smaller than 10 kilobases, we are going to exclude it. However, this is also not true in every case, since if there's enough DNA left of the sample, you can of course account for the portion of fragments that you lose in the preparation by just using more of the sample itself. On our side, we have also made the experience that bead-based extraction strategies work better than column-based extraction protocols, but this might only be true for the protocols that we've tried here in-house. Maybe as a take-home message, if you consider using long-read sequencing at all in the intermediate or even in the long term, start establishing these extraction methods now, because otherwise in 10 years, you will have very precious and rare samples, but they cannot be analyzed with state-of-the-art methods, and this would be very sad.
Thank you for your answer. I have another person asking something very interesting, which is, I have done some bead-based size selection with standard and pure XP beads for short read libraries, but why do you prepare specific size selection beads by yourself? What's the difference?
Thank you. Also a very important question. Yes, you can perform size selection with standard and pure XP beads. However, this works well for a range of around 500 base pairs. And I know that this is established in many short read library preparation protocols. However, if you want to get in the ranges that we require for our long fragment capture, which is around 10,000 base pairs, you have to exchange the buffer of the beads. You cannot do size selection in that range with the standard and pure beads. So with the buffer that we exchange for the beads, we actually have a very strict cutoff of around 6,000 base pairs in which we do size selection. And you cannot achieve this with standard and pure beads.
Thank you. Thank you for this answer. And we have one more question for you, Andre. The question is, do you think that this protocol can be applied in translational clinical research?
It's definitely of interest. So currently we are already doing routine diagnostic testing for blood donors here at our university hospital, but we're using genotyping methods. They still have the advantage, of course, of the throughput and the low per sample cost. Nevertheless, there are also samples where the blood group composition is less clear, and here sequencing, of course, provides the higher resolution. Now, in order to make this protocol amenable for clinical practice, I think there are still some improvements needed, but it's definitely the way forward. Yeah, in terms of improvements, I would say per sample cost, of course, need to be reduced even further, especially if we're going for high throughput blood donor typing. For patients, it's less high throughput, but here speed matters, and here we rely, of course, on the sequencing technologies to improve in speed.
Definitely. Thank you so much, Andre. Thank you, Tim, for this wonderful presentation.